Associate Professor Kelly Avery-Kiejda is a Cancer Institute Career Development Fellow at the Faculty of Health and Medicine School. Oh, sorry, I'm going to start that again.
Yeah, you're right. Go for it.
Associate Professor Kelly Avery-Kiejda is a Cancer Institute Career Development Fellow at the Faculty of Health and Medicine, School of Biomedical Sciences and Pharmacy at the University of Newcastle here in Australia, as well as an affiliated researcher in the Cancer Detection and Therapy Research Program of the Hunter Medical Research Institute. A highly accomplished molecular biologist who specialises in studying genetic mechanisms that enable cancer cells to evade treatment and metastasize to remote parts of the body, with a focus on breast cancer and melanoma, two prevalent and challenging to treat cancers here in Australia. Kelly's groundbreaking research has significantly advanced our understanding of the genetic drivers that underlie the progression of these diseases. Her work has shed light on the key factors that contribute to the high rates of morbidity and mortality associated with cancer metastasis and has opened up new avenues for the development of more effective treatments. Welcome to Pure Health Hub, Kelly.
Thank you so much for that introduction, and I'm really glad to be here.
Oh, I'm thrilled to have you. Now, all I wanted to do today really was chat to you, the expert, about the latest in breast cancer treatment in the treatment arena, with treatment resistance obviously being a concern. I wanted to learn a little bit more about that, but also the latest developments in helping women beat it. So you and your team have conducted considerable research into resistance of breast cancer treatment. But I understand there was a recent discovery that may help to understand why some people are resistant to treatment. Could you elaborate?
Yeah, absolutely. So basically all of the treatments that we use in breast cancer today, so we do have what we call targeted therapies, which you may have heard of, things like tamoxifen and so forth, which could be used for women who have, say, hormone receptors for estrogen receptor, etc. But other than that, they're also used alongside what we call DNA damaging chemotherapy. So most of the chemotherapies that are used today are quite general and broad, and a lot of them work by damaging DNA. And they damage the DNA in the cell so severely that it causes the cell to die. And so what we are working on is a key protein involved in this process called P53. So P53, if it isn't working properly, basically the cell won't die. It's a key mediator of events as to whether the cell will live or die. And if the damage is not so severe, it will live. And if the damage is really severe, it will die. So P53 obviously plays a big role in chemotherapy responses because it will cause the cell to die when that DNA damage hit comes. And so P53 is known to be mutated in around 25% of breast cancer cases. And this really got us to thinking, well, it's only in 25% of cases. Surely something else is going on, because this is really a key mediator of the DNA damage response. So could this be something that's happening in breast cancer and why breast cancer is not responding to therapy? And so when we took a closer look, we found that there were these smaller versions of P53. We like to call them the little brothers of P53. And we found that they were present in around 45% of breast cancer cases. And so when we looked further into this, we actually found that this was associated with the development of distant metastases over time. And so those women who had higher expression of these little variants, they fared worse to chemotherapy treatment over time. And so we've now gone on to have a look at that in breast cancer cells in the lab to see what happens if we can try to inhibit these smaller variants. Will that perhaps turn P53 back on and make the cell die? And so what we've found is exactly that. So if we can inhibit these smaller variants, we can then turn this, the DNA damage response back on, and the cells are more sensitive to these DNA damaging chemotherapies. So this basically looks like it could be potentially, I guess, a therapeutic response if we develop it further to increase the response to therapies in cases where we think that P53 might not be causing that appropriate cell death response to the chemotherapy.
Wow, that is extraordinary. That is extraordinary. So, you're constantly flipping it. You're finding the cell and it's not performing. Okay, so is treatment resistance something that you establish instantly? Like, is someone who's resistant automatically resistant, or does this resistance happen over time? And then what are the options if this happens?
So there are a number of theories as to how resistance develops in people, and quite honestly, it could be both. So your tumor, so there are many factors that actually affect the drug response, and this could be drug metabolism, activation or inactivation of certain drugs, drug responses, elimination of like the elimination mechanisms of the tumor. It might eliminate the drug too quickly so that it doesn't work. There might be changes in the targets that the drugs are actually activating, as I mentioned. So there could be so for instance, in breast cancer, tamoxifen, that's known to basically work alongside the estrogen receptor. So there could be mechanisms blocking that pathway as to why patients aren't sensitive to tamoxifen. So there's a lot of mechanisms like this. There could also be overexpression of proteins involved in DNA repair, so that the cell is deciding to try to repair the DNA rather than die. Lots of these things that could actually be present within the tumor initially. So they could be there. But there's also something that we call, or it's well known, I guess, in the science field, called the stem cell theory. And so we believe that there is your tumor is made of obviously thousands and a big mass of cells. And so basically, we don't believe all of those cells are resistant. So your cancer therapy will come along and it might, kill off most of those cells, but then you'll have a few of these cells which are really resistant, and we call them the cancer stem cells. And it's then once all those cells that have responded to therapy die, these stem cells will then grow, proliferate, and they can't be killed by the chemotherapy. So there's two theories, I guess. There could be innate mechanisms that are in the tumor itself, and so you could actually, be resistant first up, or it could be something that develops because you've already killed off those cells. The tumor looks like it's responsive, but if it hasn't killed off all of those cells, then you're going to get more resistance. And so what are the patient's options if that happens? So generally, it's going to depend on how I guess how far along the patient is, to be honest, and how, if the tumor is too big or if it is metastasized to multiple areas. But generally, and also what the patient wants in terms of quality of life as well. So
Yeah.
that's also a big factor. But I guess a lot of the time it is just treating with blunt chemotherapy drugs, again, which are more likely to damage DNA, or looking for things that once the tumor has been analyzed, if we know that there are certain proteins or genes that might be mutated or not mutated, we could maybe use targeting drugs. But generally at the moment, it's very broad agents and potentially combination chemotherapies to try and target other pathways that are activated within the tumor. That's generally the options, just generally chemotherapy, very blunt.
Yeah. So you go back to the hard hitting,
Yeah, there's nothing really targeted at the moment. It's very unfortunate.
Yeah. But you're working on it, right? So
yeah.
So is it a type of breast cancer that becomes resistant, or is it a person's innate biology? I mean, you sort of explained could be either, but do you think it's more about their innate biology?
So look, there are definitely, if we look at breast cancer, there's a whole bunch of subtypes of breast cancer, and they can be classified through a number of mechanisms. A way that they're commonly classified is by their hormone receptor expression. So hormone receptors that we generally look at when we type a tumor would be estrogen receptor, progesterone receptor, and HER2. And so these are important because both ER and HER2 can be targeted, and that's why we look at these. So this is one of the most personalized therapies, I guess, that we get in breast cancer, is looking at those. And if you can be treated with hormone or anti-hormone agents, we'll do that. So in terms of whether a tumor will metastasize, more likely there is, like, the HER2 positive tumors tend to be more aggressive. So around 50% of those can acquire resistance to therapy. And also there is a subtype called the triple negative breast cancer subgroup, which is basically negative, doesn't have ER, estrogen receptor, a progesterone receptor or HER2. And so they are quite aggressive and quite unresponsive to chemotherapy. Or, well, they respond initially, but then they are more known to develop resistance later. So it is definitely something that can be dependent on the type of cancer you have. Yeah, I guess it also depends on, the aggressiveness of it. And as I mentioned before, so yes, it could be depending as well on if there are drug metabolism mechanisms or genes that are known to be mediating the effects of the drugs. If they're potentially overexpressed, so highly expressed, or underexpressed, then that will also affect the drug response. So it can really be both. And as I said, the cancer stem cell theory is the big one, and that's a big thing that we work on actually, is we believe so that these little variants that I mentioned at P53, we actually believe that they are present in the stem cells, and we think this could be a key reason as to why they are affecting chemotherapy response as well.
Right. And just briefly, is it the same for men and women? Like it's the same treatment?
I'm going to say yes. We don't really work in male
No.
breast cancer because it is such a small proportion, and that's really bad to say because we should.
I know, it's just how many geese you're going to kill at once.
Yeah.
Okay. Yeah, no, I was just thinking about that then, because obviously our hormones are quite different, but yet the same. So I don't, it's all interesting.
So I can't actually think as to whether I can't. I'm not sure whether they express the estrogen receptor in their tumours, to be honest, because they don't obviously,
All right, this is all about women. So, but it just came to me, and I thought, all right, now given the depth,
it's a good one though, because I should know that. Yes.
Sorry, I shouldn't have put you on the spot.
No, it's completely fine.
Now, given the depth of your research, could you tell us where we're at in this cancer battle, and what is the future focus on global breast cancer research? I know that's a big question, but from where you sit.
I think we've actually come a long way in terms of breast cancer. I mean, and it's one of the good cancers to get, to be honest. I mean, there's a 92% survival rate,
Wow. Okay, that's good.
We can actually, treat it fairly effectively. If you catch it early, and obviously it's very subtype dependent, it has a 92% 5-year survival rate. So that's fairly good when we compare to other cancers. It is one of the most prevalent cancers, and it's certainly the second highest cause of cancer-related deaths in Australia. So it's not one that we should go, we're done with, because we absolutely have a lot of work to do in there, which is, what we're focused on. So what is the key focus at the moment for breast cancer researchers? Basically, the key focus is trying to target metastatic breast cancer, because what we know is breast cancers are fairly responsive to treatment, but once they become resistant or they go to secondary sites, that's when they become unresponsive. So what scientists are generally looking for is key markers in the cancers that can basically say whether this cancer is going to metastasize, or if it has metastasized, what's one of the best targets that we could use to kill this cell off. So that's one of the main things. So we would call that sort of more personalized therapies. So we have the ability to look at the genomic profile of these tumors, and so what scientists like to do is to profile these tumors, and we can even do it at a level at the single cell. So we can look at every cell and see, and it's crazy how remarkably heterogeneous these cells are. They're all very different within a cancer. It's not like your cancer is completely the same. So this is one of the reasons that metastasis and treatment resistance develops, because they're not the same. So you have to try and pick what's the good cell, what's the bad cell. And so that's what we're really trying to discover at the moment, which cell's the bad one that's going to become treatment resistant or metastasize. And so how do we personalize therapies? So that's one of the key things that we're doing. And the other thing is there is a really big focus on this triple negative breast cancer subtype, as I mentioned. So they are one that has no targeted therapies at the moment, and they are quite aggressive. And so people are really trying to look at targeting therapies towards that subgroup.
Wow, it is just so complex, right? When we hear about cancer, and we know there's various cancer, we have ovarian and we have breast and we have, all the others. But within those breast cancers, there's another whole world going on. There's all the verticals
It's
within that.
all
So,
cancers too. It's not just breast cancer. Pretty much
yeah,
all cancers are very heterogeneous, and so that's one of
yeah,
the biggest problems and one of the biggest
challenges.
challenges that we're
Sure.
trying to overcome. Yeah.
Sure. Well, you know what? You really are the unsung heroes, seriously. And thank you so much for today, but also thank you for what you do. I know that a lot of women are very grateful for that. So thanks, Kelly. Thanks for joining us today.
Thank you so much for having me.
Pleasure.
Thank you.